All right, welcome Science 10. This next lesson will be regarding microscopes. So how do scientists visualize the microscopic world? Obviously through microscopes. There's many types of microscopes. We'll take a look at different types today. All right, pictures like this are not possible without microscopes. This is one of my favorite organisms ever. This is called a tardigrade, and they're obviously quite insanely ugly. You'll never see one in the world. They live in moss. Why they're super cool to me is because they are most likely space travelers. Yes, they travel through space. All right, evidence of them has been found on asteroids. Oh my goodness, how'd they get into space? Well, if an asteroid were to hit the planet like it did in the past many times, even while life was on the planet, and chunks of rock get spewed out into space, all right, these guys have the ability to go into a kind of stasis, a kind of like lifeless state, and just like pause their little lives. And once they get back into an environment that can support them, mainly water, okay, then they'll reanimate, which is super cool, right? Which happens to moss all the time. Moss dries out. These guys have to go into their stasis. They wait until the moss gets wet, and there they go. Anyway, this lesson is not on tardigrades, although they're super cool. I wonder if you know what these are. Oh my god, I don't believe I put those in there. Okay. Yeah, and tadpoles, obviously. Those are ova in vitro with sperm covering them. Oh my goodness. I don't know why I picked that. Okay, let's keep going. So compound light microscope is what we use in our classes, and we're going to be learning about compound light microscope calculations. Normally we would do a lab. Okay, but why do we call it compound? Because they have two lenses. Okay, so that means the compound and light. They use light obviously to visualize the specimen. Okay, before I keep moving, there's a web quest for you to do on all these microscopes, so I'm not going to take very much time at all with this. I'm going to let you work through the web quest and learn what you need to learn about them. So parts and images of the compound light microscope, again, you're going to have to go through this on your own and identify all the pieces. Some of the more important things, eyepiece, which is a magnifying lens. This is a 10 times magnifying lens. And the objective lenses. Normally a microscope will have 3. In our labs we have 3 objective lenses. And there's a 4 times, a 10 times, and a 40 times objective lens. Now, the total magnification is we have an eyepiece at 10 times, let's say, and an objective of 4 times. So we have to multiply those together, and we'll get to that. Okay. The rest of the microscope, please go through and learn the different parts and their functions, please. So there are types of electron microscopes. There's transmission, scanning. Oh, where's the other one? And scanning tunneling. There it is. So what's the difference between these microscopes? On a simple, all right, for a simple lesson, TEMs, all right, they're like light microscopes. They operate on the same principle. You need a slide, you need a slice of specimen, and the electrons will pass through the specimen right here, okay, and the person can observe it with their eyepiece. So again, the electrons get passed through the specimen and the image will come to the viewer. Whereas in a scanning electron microscope, which are my favorites, you're scanning the surface of objects. Okay, this is, I don't know, oh my god, I think this is a grain of sand, if I remember correctly. It very much could be a grain of sand because these are all types of diatoms which only exist in oceans. These are ocean algae, or these are the bodies of them. So this would be silica. Okay, this is where we get silica from, is diatoms. I think this is the surface of your body. So you want to keep looking at that, right? You're like, what are those crazy creepy crawlies there? Like, I don't know what they are, but oh my god. Yeah. So we're covered. You are ecosystems people. Get used to it. Okay, and for the scanning tunneling microscope, this is a very cool microscope. Look at it, it's insane, hey? Now, what is this thing able to do? It's able to picture atoms. You can view atoms with these, all right? And instead of, right? And so with this microscope, the point of focus is actually one electron, right? Whereas with the scanning electron and the transmission electron, it's not just one electron passing through the specimen, or in this microscope, reflecting off the specimen. In this microscope, this is one electron, all right? So it can have a very, very, this is the most microscopes that we have in the world, meaning that it has the most magnification of any other microscope, obviously, because we can view atoms with this microscope. This is a cool microscope because the confocal laser, because it can take 3D pictures and produce them on a computer screen that you can actually rotate the specimen around. You can go into the specimen, I believe. Yes. All right. So this is a cell membrane. This is a really an amazing picture of a cell membrane. And obviously we're not going to tell you what the parts are. And these are just, I don't know what kind of cells these are. These aren't eggs. These aren't red eggs. They're cells. So again, go through your web quest for microscopes. It's part of your assignment today, and I'm going to move on here. We're going to skip areas of cellular research in this course, so cross that out. And last thing we do today are viruses versus prions. Now, I would have also scratched this out. However, we're at home because of a virus, so let's take a look at viruses a little bit here. Okay, I have a great video here. Please watch it. It's the coronavirus explained. It's a great video. It's not boring at all. Okay, some of them are, but you should be watching all the videos I'm posting on your PowerPoints. Yeah, they'll give you more in depth than I will here. Virus, a microparticle. Remember, it's not alive. Smaller than a bacterium. It's about 100 times smaller than a bacterium. Okay, that can grow or reproduce apart from a living cell, or cannot. There, I'm like, what? Can? No, good. Cannot grow or reproduce apart from a living cell. So what we have now, this is a bacteriophage. These only will infect bacteria. Now, all viruses, whether they're bacteriophages or this is the HIV, this looks very much like coronavirus. Okay, so most of the viruses that infect us are round in shape. They have a protein body, and they have a piece of DNA or RNA or genetic material inside of them. Same with the bacteriophage, there's DNA or RNA within the phage. And what they do is they attach themselves to cells. Bacteriophage will hook onto a bacterium. These are more specific. They hook onto specific receptors on our cells, which makes them very interesting. Why do they use our cell receptors? How do they know our cell receptors? This is all in research right now. I think we understand very little about viruses at all. Okay? But they'll attach to the cell and they will inject their genetic material into the cell. The cell will then make new viruses, new virus particles, and the virus particles will explode out of the cell. Why do we feel sick? Because our immune system makes us feel sick. Okay, and we want to be very clear about that. Watch this video and it'll explain why we have symptoms. Otherwise, we should not have any symptoms because we're not actually being harmed too much by them. They're going to take out some cells. Okay. Prions, we're not going to go into in Science 10, so you can, if you're interested, great, go ahead, learn about them. If you're not, good enough. All right, thank you. That's the End of this lesson, and I'll see you in the next lesson for microscope calculations.